Hello and welcome back. Today we will be covering the pentose phosphate pathway. So the function of the pentose phosphate pathway, I will be referring it to PPP from now on, is that it is important for the red blood cells because it occurs in the cytosol and is a significant mechanism to produce NADPH. And remember, red blood cells don't have any mitochondria, so they are reliant on only anaerobic metabolism. So this is also very important in the liver, mammary glands, and adrenal glands because in these structures and organs, this is where fatty acid synthesis occurs. So the general gist of the PPP is the direct oxidation of glucose to carbon dioxide and releasing two NADPHs. The function is to create ribose 5-phosphate and NADPH, and it also plays a role in protecting the cell from free radicals. The ribose 5-phosphate is created for the nucleic acid synthesis, and the other products created, the fructose 6-phosphate and glyceraldehyde 3-phosphate, will go into glycolysis. The PPP is set up into two distinct phases, the oxidative phase and the non-oxidative phase. The first phase, the oxidative phase, is all of it is basically irreversible, and they're all kind of committed steps because of the irreversible steps. So let's start out with glucose 6-phosphate, and using the glucose 6-phosphate dehydrogenase, it uses an ATP to make 3-keto-6-phosphogluconate, which is the intermediate between these two. And from there, it also creates an NADPH. And then oxidative carboxylation uses another ATP, and then tautomerization to make the final product, ribulose 5-phosphate, along with carbon dioxide. So these few steps will create two NADPHs. So the hydrolyzation of G6P to 6-phosphogluconate is considered irreversible, but in vitro it is reversible, but in the body there's no such way for the pathway to go backwards. This is the overall reaction, as you can see here, glucose 6-phosphate, and then two NADP pluses, and then water. It eventually makes the ribulose 5-phosphate along with two NADPHs, 2 hydrogen, and 1 carbon dioxide. Because of the carbon dioxide produced, this makes it essentially irreversible. The non-oxidative phase is basically just a reshuffling of the carbons by either the transketolase enzyme or the transaldolase enzyme. And this process is reversible so that the body can make the ribose 5-phosphate without making the NADPH, or in some variety of other ways. We will cover that in the slide about regulation. But what to know, the transketolase will transfer 2 carbons while the transaldolase will transfer 3 carbons. And eventually they all make the glycolytic intermediates, fructose 6-phosphate. And then, of course, that can go into glycolysis. As you can see here, glucose, then this will be, broken down into G6P. And then from there, this would be the PPP, and here, ribulose 5-phosphate, and then it can go into either this, ribose 5-phosphate, or xylulose 5-phosphate. And then that can be transketolase will create the glyceraldehyde 3-phosphate, and then from there can make the erythrose 4-phosphate, and then with another reaction, it can also create G3P or F6P, and then those can go into the glycolytic cycle. Then from here, the ribose 5-phosphate goes through a transketolase into the cetoheptalose 7-phosphate, and then it can be it has a transaldolase to transfer an aldehyde into fructose 6-phosphate, and then that can go into the glycolysis, which is a long and kind of complicated way of saying, like, there are these two pathways, and eventually they will make the glycolytic intermediates. This one is a little bit more straightforward. This one just has an extra step, and this is the overall reaction. Sorry, there you go. So as you can see here, you start out with the 3-ribulose 5-phosphate, and then that has to be turned into ribose 5-phosphate, and then that can ribulose 5-phosphate makes 1, or like 3 of these ribulose 5-phosphate makes 1 ribose 5-phosphate and 2 xylulose 5-phosphates, and those then can create 2 fructose 6-phosphates and then 1 glyceraldehyde 3-phosphate. Okay, let's recap all of that. So before ribulose 5-phosphate can go into the non-oxidative phase of the PPP, it has to be turned into ribulose 5-phosphate first, right? So first, this begins with the glucose 6-phosphate, and we get that from glycolysis using hexokinase, remember? And then with a dehydrogenase, that is dehydrated, and NADP is reduced into NADPH. There we go. We turn it into 6-phosphogluconolactone, and there water is added. Using this enzyme, it's turned into this, and then another dehydrogenase is used, and then this is the step that produces the carbon dioxide. What to know about these? It's important to know this enzyme in particular. It is highly regulated, and that these two steps create the NADPH. Okay, so finally you get the ribulose 5-phosphate, and then from there it can go turn into the fructose 6-phosphate directly, or it can be turned into the xylulose 5-phosphate, and then with a bunch of shuffling eventually make the fructose 6-phosphate or the glyceraldehyde 3-phosphate. Okay, so beriberi affects the PPP because if you can recall about the vitamins lecture, beriberi is caused by a lack of thiamine, or B1. So the cofactor of the transketolase is thiamine diphosphate, and that creates a problem because the transketolase connects the PPP to glycolysis. As I was waffling on before, you can see like these two cycles are very, very linked together because the PPP creates the NADPHs, right? And then those glycolytic intermediates. Okay, so the end products makes 6 carbon dioxides and a pentose per 1 G6P, and it also creates like 12 NADPHs. So 5 fructose are recovered per the 1 glucose that is degraded, and we require the 12 NADP to make the NADPH and water. So the PPP is need-based, and G6P DH, the dehydrogenase, is allosterically inhibited by high levels of NADPH and the long-chain fatty acids, especially the omega-3 fatty acids. If ATP is needed, the G6P will go directly into glycolysis. If NADPH is needed, G6P will go into the PPP. So if ribose 5-phosphate is needed but not NADPH, the cell, this is only in cells that are dividing, the ones that need to make the nucleic acids in order to duplicate the DNA. In those cells, the non-oxidative phase occurs. And then if both R5P and NADPH are needed, the oxidative phase will occur, and that will make the one R5P per 2 NADPH. And another scenario, if we need more NADPH than R5P, this generally occurs in the liver because it is going to make the fatty acids. One G6P is fully oxidized into a carbon dioxide. And if you need NADPH and ATP, the oxidative phase along with glycolysis will occur, and that makes a total of 10 ATP. So all in all, a fairly effective and efficient cycle. As you can see, all of these are very connected. So I didn't cover glycogen metabolism quite yet, but as you can see, all of these are all of these have an intermediate that can go into another cycle, and it's important to know which parts of the cycles are regulated, because that is where the intermediates are. So NADPHs are important because it plays a role in peroxide detoxification. Oxidation is damaging because it can cause cancer by the modification in the DNA and apoptosis and cellular damage. NADPH is important for the mitochondria and cytochrome C, and the body plays a role in reducing oxidative damage, not stopping it completely, because we do need some oxidants, such as phagocytes. So the phagocytes will use the NADPH oxidase to create the superoxides in order to kill pathogens or defective cells. Complex 1 and 3 of the mitochondria, if you can recall about the ETC, it will make the superoxide as a byproduct. Okay, so glutathione is an antioxidant. It uses NADPH to reduce the electrons, and it is made out of these 3 amino acids: glutamate, cysteine, and glycine. So the sulfur of the cysteine will be oxidized, so reduced glutathione will be oxidized rather than other cellular components, and it protects the other tissues in this way. So glutathione is our sacrificial lamb, if you will. NADPH reduces glutathione to remake the reduced glutathione using this enzyme, creatively named glutathione reductase. As you can see here. Here it is. This is our superoxidant, also known as hydrogen peroxide. And we will turn it into water, which can be excreted. So there is an X-linked mutation in the gene coding for glucose 6-phosphate dehydrogenase, and the low levels of this enzyme lead to less oxidative phase, right? So this doesn't occur as much, which decreases the NADPH levels. With the low levels in the NADPH, glutathione can't be reduced or re-reduced, as you can see, it's recycled, right? So without the reduced glutathione, oxidants can't be detoxified, and they can come into the cell and damage the red blood cells. The low amount of reduced glutathione compromises hemoglobin in the red blood cells and causes them to cross-link, making these Heinz bodies, which deform the cells, and the deformed cells are noted by the immune system and are lysed, causing the hemolytic anemia. This is linked to people of African and Mediterranean descent, and by the Mediterranean I mean people who have like Greek or Turkish or Albanian heritage. There is a role to make these populations resistant to malaria, because malaria is a parasite, and by lysing the cells, it doesn't allow the malaria parasites to mature within the cell and then use the cell's mechanism to create more malaria parasites. And by lysing it early, it essentially like kills the malaria while it's in its like whatever part of the life cycle it is in. So it's also caused by oxidative drugs, fauvism, which is in people with a G6P deficiency can be induced into a crisis by eating fava beans. It's also caused by like infection and jaundice. And the oxidative drugs, such as the antimalarials, those are like quinones, and those increase the superoxide activity. That's it for now. Thank you for your attention, and good luck studying.